This study presents DRAVA, aligning human concepts with machine learning latent dimensions, for the visual exploration of small multiples. Small multiples are commonly represented as latent vectors and then be visualized in a scatter plot using dimension reduction. Even though revealing interesting patterns, this method can be hard to interpret. We propose DRAVA to address this issue. DRAVA is a visual analytics system that arranges, groups, and summarizes items using interpretable concepts, which are learned without human supervision. In the concept view, each row represents a latent dimension and is ranked by saliency scores. Users can interpret a dimension based on the synthesized images, and rename it to facilitate the following analysis. Rows can be added or deleted as needed. Users can further verify the concept associated with a dimension by changing the baseline image. In the item browser, users explore the small multiples by arranging, grouping, and summarizing them based on these semantic dimensions. The spatial view is an optional view for data items that have spatial information. It is coordinated with other views to reveal the correlations between concepts and item context. Here, we demonstrate the effectiveness of DRAVA through 3 usage scenarios. In the first scenario, each item is an image patch of breast cancer specimens. Users first use UMAP to arrange all items, but it is hard to interpret. Therefore, users arrange items based on 2 semantic dimensions, one is related to the tissue density and one is related to the tissue color. Users can see a strong correlation between the presence of cancer cells with these 2 concepts. Users further filter items to investigate confident false negative predictions, finding that these are mostly loose purple tissues, that is, fat tissues surrounded by cancer cells. In the second scenario, each item is a region of interest, called TAD, from a large genomic interaction matrix. Users can identify 3 interesting dimensions that correspond to important attributes of TADs and indicate specific biological events. However, when users group items based on a dimension that is related to the nested structure, the item previews reveal abnormal items, indicating that the backend model confuses the thickness of the diagonal with the nested structure. To refine this concept, users use another dimension as the y-axis to help separate nested TAD from single TADs with thick diagonal. Users then select all single TADs that have large dim_nest values with a lasso selection and move them to the leftmost position. This refinement is recorded using the local update mechanism and applied to similar items. In the third scenario, items are shapes with different rotations and scales. Users can see that UMAP puts items with the same shapes and scales together, but overlooks the position of shapes. This issue can be addressed by arranging items based on 2 position-related latent dimensions. Users provide group summary and allow users to examine individual items. Users investigate items based on a dimension related to the scale of shapes. Only squares are on the left side, indicating a backend model fails to learn the concept scale properly. Users can provide feedback to fine-tune this concept. By choosing SDD as the y-axis, users identify some large ellipses that are mixed with medium squares. Users modify the position of these items, group items, and initialize a concept adapter. This concept adapter can be progressively tuned by splitting and merging groups. And reassigning the groups of individual items. Our experiment shows that user refinement can effectively improve the quality of concepts. Thanks for watching. You are welcome to read our paper and visit our website to find out more details.